Hello everyone, today we are going to discuss our next chapter number 2, that is microorganisms, friend and foe. This is your class 8 topic, and today we will study what are microorganisms and how they act as your friends and how they act as your foe. So they can be benefited also, and they are not good for us also. So how this we will study now. Now first is your introduction, that is what are microorganisms. So the microorganisms, which are also called microbes, why they are called so? Because they are very small, they are in micrometers and they can't be seen by our naked eye, and we can see only through microscope. So that's why they are known as microorganisms. The microorganisms or microbes are so small in size that they cannot be seen with the unaided eye. Unaided eye means the naked eye. Some of these, such as fungus that grows on bread, we all know, can be seen with a magnifying glass. So what is a magnifying glass? Magnifying glass is a glass which is used to magnify the objects, which is used to zoom the image, and we can see that object very clearly. So others cannot be seen without the help of a microscope. That is why these are called microorganisms or microbes. Microorganisms are further classified into 4 major groups. So the microorganisms, they are of 4 types. They are divided into the 4 groups, and that groups are: first is your bacteria, second is fungi, third protozoa, and fourth is some algae, that is blue-green algae. So we will see this picture. You can see these are some of the microorganisms. These are very small, and now we will see one activity which is related to your study. That is, you can perform this. You have to collect some moist soil from the field in a beaker and then add water into it. After the soil particles have settled down, because after adding water to the soil, your sedimentation process will take place, and the heavier particles, which are soil, they will settle down at the bottom and the water will go up. So you have to observe a drop of water from the beaker under a microscope. You will take some water from the beaker through dropper and then put on a slide and observe it under a microscope. What do you see? Please give your comments or answer in the comment box, that what will you see and what will you observe. Now coming to activity 2, this you can also perform very easily. You have to take a few drops of water from a pond, then spread on a glass slide and observe through a microscope. So what you will observe? You will find very tiny microorganisms which will be seen moving around the object. So this microscope, it is used to magnify the image, and it is able to see the images of your microorganisms which you can't see by your naked eye. So these are some of your observations. Those tiny things which you observe through the microscope, they are known as microorganisms. Some of the pictorial examples are given in your next section. So you can see these are the different types of bacteria. The bacteria have different type of shapes, and those shapes can be your spiral bacteria. You can see the first picture. These are spirally shaped. Second one is your rod-shaped bacteria. These are in the form of long rods. So these are rod-shaped bacteria. And many types of bacteria are also there. For example, Vibrio, that is comma-shaped bacteria, and Bacillus form of bacteria is also there. So mainly 4 shapes of bacteria we have studied. So coming to next. Now these are the types of algae which you can observe. First is Chlamydomonas. This is the image how Chlamydomonas looks. And second is your Spirogyra. So These are the two examples of your algae. So, what type of reproduction takes place in Spirogyra? Please comment in the box that which type of reproduction takes place in Spirogyra. It is sexual or asexual, and of which type? If it is asexual, then which type? Please comment down. Okay, coming to next topic. These are the types of paramecium. Fine, we have studied the bacteria shape, and then we have studied about algae shape. Now next is your paramecium. First is your amoeba, second is paramecium. So you have studied about amoeba a lot in your previous classes also. Amoeba is a microorganism which is unicellular. Why unicellular? Because it is only single cell, only single type of cell is present in amoeba, and moreover it is immortal because it can never die in its whole lifetime. So this is amoeba. It undergoes your binary fission also and multiple fission also in case of asexual reproduction. And second is your paramecium. Now next is the types of fungi which you can see clearly in the picture. First is your bread mold, second is penicillium, and at the top conidias are present and penicillia are there. Third is your aspergillus. So these are the examples of your fungi which can act as your microorganisms. Next is some types of viruses which are also found. So these are the two types of viruses. Viruses can be present in many organisms. For example, your bacteriophage is also one kind of virus which acts on the bacteria. So viruses in human body, they cause many type of diseases also. So they mostly act as your foe. So these are the viruses which are shown in the figure. Okay, so now next is where do microorganisms live? Microorganisms, they may be single celled also, that is your unicellular. Examples can be bacteria, some algae and protozoa, or they can be multicellular also. Multicellular means, multi means many and cellular means cells. So the organisms in which many cells are there, they are known as your multicellular, such as algae and fungi. They can survive under all types of environment, ranging from ice cold climate to the hot springs and deserts to marshy lands. So the microorganisms, they can survive in all kind of habitat, all kind of temperature conditions. They are also found inside the bodies of animals including humans. Some microorganisms grow on other organisms, so while others exist freely. So there are some kind of organisms also which can grow on other organisms, such as your parasitic host relationship. And second is where the organisms, they exist very freely. So microorganisms like amoeba, which can live alone, while fungi and bacteria, they live in colonies. They can't live alone, they make groups and they make colonies. So these are the some type of habitat of microorganisms and where they live. Coming to next is what are friendly microorganisms. So our chapter is mainly about friend and foe. So now what are friendly microorganisms? Microorganisms are used for various purposes. We all know. You can also comment in the comment section box that please think carefully and think where the microorganisms, they are useful to you at your homes. Please comment down. So microorganisms are used for various purposes. They are used in the preparation of curd. You all know how curd is prepared from milk. Fine. In milk we add inoculum from the curd. Inoculum means a pinch of curd we add to the milk after heating it, and that inoculum contains millions of bacteria. And which type of bacteria is present in curd? The name of bacteria is your lactobacillus. So lactobacillus is the bacteria which helps in the preparation of curd from milk. So it is a useful microbe for us. Next is bread. Bread also, in preparation of bread is fluffy and soft. So what makes it fluffy and soft? Your yeast helps in the fermentation of products, and we get bread and other bakery products. So they are also used in cleaning up of the environment. Microorganisms also clean our environment. For example, the organic waste such as vegetable peels, remains of animals, feces, etc. They are broken down into harmless and usable substances by bacteria. Recall that bacteria are also used in the preparation of medicines. They also make medicines. In agriculture also, they are used to increase soil fertility. And how? By fixing the atmospheric nitrogen into various nitrogenous forms. So bacteria are useful to us in many ways. Next is your making of curd. You can see in the picture also, curd is prepared from milk. How curd is prepared? Curd contains several microorganisms. I have told you about inoculum. It contains several microorganisms called bacteria. Of these, the bacterium lactobacillus promotes the formation of curd. It multiplies in milk and converts into curd. Bacteria are also involved in the making of cheese, pickles, and many other food items. An important ingredient of rava, that is suji, idlis and bhaturas, is curd. Because in making rava, idlis and bhaturas, you all have noticed that we add curd into it, so that it can make it fluffy and we can eat it very lovingly, and it tastes very delicious. So it is the usage of these bacteria we are studying. Next is making of bread. So how bread is prepared? The bacteria, the organism that is present in the bread, it is called yeast. So yeast reproduces rapidly and it produces carbon dioxide gas during respiration. Because of this carbon dioxide gas only, the material which we are going to eat, for example your cake, bread or pastries or any other bakery product, it becomes fluffy because of these carbon dioxide gas. So bubbles of the gas fill the duff and increases its volume. This is the basis of use of yeast in the baking industry for making breads, pastries and cakes. So these also act as your useful microorganism. Coming to next activity, you can do this. First is your maida with yeast powder. Second is your raised maida. Why your maida is raised afterwards? Because of the presence of yeast in it. Yeast makes your material fluffy and soft. So you have to take half kg of flour, atta or maida, and add sugar and mix with warm water. Not very boiling water, only warm water. Add a small amount of yeast powder and knit to make a soft dough. What do you observe after 2 hours? You have to tell what will you observe after 2 hours. Did you find the dough rising? Please observe at your home also when your mother cooks something for you, for example your cake or any other material, that dough which your mother have prepared, it rises after some time. After 15 to 20 minutes it rises. Why it happens? Because of the yeast in that material. That yeast organism, it helps in the raising of dough, it helps in the fermentation of that material. Now observation. Yeast reproduces rapidly and produces carbon dioxide during respiration. Bubbles of the gas fill the dough and it will increase its volume. This is the basis of use of yeast in the baking industry for making breads, pastries and cakes. So this I have already explained you. Now next is commercial use of microorganisms. What is the commercial use? Now microorganisms, they are used for the large scale production of alcohol in alcohol industries, then wines and acetic acid. Acetic acid is your vinegar. Acetic acid, what is the formula of acetic acid? If anybody knows, please comment down. Yeast is used for the commercial production of alcohol and wine by the process of fermentation. For this purpose, yeast is grown on natural sugars present in grains like barley, wheat, rice and crushed fruit juices, etc. The process of conversion of sugar, first sugar is there as a raw material, then fermentation of sugar is done. After its fermentation we obtain alcohol or wine, and this process is known as fermentation. Okay, now coming to activity 4, you have to take a 500 ml beaker. You can see in the picture, this is a 500 ml beaker, and you have to fill it with about 3/4 of water. Then dissolve 2 to 3 teaspoons of sugar in it and add half a spoon of yeast powder to the sugar solution. Means you have to take water in beaker, then add sugar into it, and after that add yeast powder into it. Then keep it covered in a warm place for at least 4 to 5 hours. Now you have to observe it, and after that you have to smell that solution. Will you get any smell? So you have to observe it, and what you will get, this is the smell of alcohol. You will get the smell. You will get a different smell as the sugar has been converted into alcohol. You know the smell of sugar, that is very sweet, I find. But when the fermentation of sugar will take place, its smell also changes, and you will not notice it very easily. So the smell of alcohol is there. And how alcohol has been prepared? By the action of your yeast. This process of conversion of sugar into alcohol is known as fermentation. Now next is medicinal use of microorganisms, where the microorganisms are finding its application in the medicinal field. So whenever you fall ill, the doctor may give you some antibiotic tablets, capsules or injections such as of penicillin. So whenever you fall ill and sick, the doctor gave you tablets, you have noticed, and that tablets, capsules or injections are of penicillin sometimes. So penicillin is also a microorganism which was discovered as it finds medicinal use for us. The source of these medicines is microorganisms. These medicines, what they do, what is the action of these microorganisms? These kill or stop the growth of disease causing microorganisms in our body. So this is the reason that these medicines, they are known as your antibiotics. These days a number of antibiotics are being produced from bacteria and fungi. For example, your penicillin is first, then second is your streptomycin, then tetracycline and erythromycin. They are some of the commonly known antibiotics which are made from fungi and bacteria. So in this way, these organisms, these microorganisms, they are finding medicinal use. Now the antibiotics are manufactured by growing a specific microorganisms, and they are used to cure a variety of diseases also. Antibiotics are even mixed with the feed of livestock. You all know about the feed of livestock and poultry to check the microbial infection in animals. So these microorganisms, they are also finding their use in treating the infections in certain animals also. They are also used to control many plant diseases. Now next is about Alexander Fleming and penicillin. Now if I ask you that who discovered penicillin, so it was none other than Alexander Fleming. In 1929, Alexander Fleming was working on culture of disease causing bacteria in his lab. Now suddenly what happened? He found the spores of little green mold in one of his culture plates. Now if he found something different, he realized that why it happened so he observed that the presence of mold, it prevented the growth of bacteria. In fact, it also killed many of these bacteria. From this, after observing this, he thought himself that why this happened so. So in this way, the mold which was there, he named it penicillin, and in this way the penicillin antibiotic was prepared. Now next is Edward Jenner and smallpox vaccine. You all know about smallpox disease, which has been eradicated now, but in previous times a disease was there which was caused by the virus, that is your smallpox, and in later stages a vaccine was also discovered for smallpox, and that vaccine was discovered by Edward Jenner. So Edward Jenner discovered the vaccine for smallpox in 1798. Now it is important to remember that antibiotics should be taken only on the advice of qualified doctor. You already know this, that you should not take any medicines without the prescription of any qualified doctor. So also you must finish the course prescribed the doctor. Whenever you fall sick, you go to hospitals or you go to clinic, and the doctor gave you medicines also. So that medicines, the course that the doctor has given you, you have to complete that course. If you take antibiotics when not needed or in wrong doses, fine, so it may take the drug less effective when you might need it in future, fine. Let us suppose if you are not having serious illness and still you are taking medicines daily, so you have to stop this, because taking medicines daily without any proper prescription, it can cause harm to your body whenever in future you will fall for that disease. Also, the antibiotics taken unnecessarily may kill the beneficial bacteria in the body, and in this way antibiotics are not effective against cold and flu, as these are caused by viruses, because your body immune system now it is not very strong because of the act of that antibiotics which you have taken unnecessarily. So you have to take care of these precautions very seriously. Now next is, why are children and infants given vaccination? You have listened at most of the times that vaccines are given to children at different ages. So why vaccines are there? When a disease carrying microbe, it enters your body, the body produces antibodies to fight against that disease, that invader which have entered into your body. So the body remembers. Now first time the body knows that who entered. Now for next time your body knows that how to fight with that microbe when it will enter again into your body. So if dead or weakened microbes, they are introduced in your healthy body through vaccine injection, the body fights and kills them by producing suitable antibodies. So in this way, after the production of antibodies, these remain in your body, and in this way we are protected from the disease causing microbes. And this is the process which is known as vaccination. This is how a vaccine works. So several diseases such as cholera, tuberculosis, smallpox, hepatitis can be prevented by taking vaccine. Now, in your childhood, you must have been given injections to protect yourself against several diseases. You have observed also that when you are infant, when you are a small child, many injections are there which are prescribed by the doctor. Many vaccines are there. So, this is just to protect you in later stages of life from certain diseases. Can you prepare a list of these diseases? So, you please comment down that what are the diseases that are there for which you have to take a vaccine. You may take help from your parents also, because you might not be much aware of that. So it is essential to protect all the children against these diseases. Necessary vaccines are available in the nearby hospitals. You might have seen the advertisements also on TVs, newspapers and magazines also regarding the protection of children against polio. Polio is a serious disease. The advertisement is there under the Pulse Polio program. So vaccines are also available for polio disease also. So this is about vaccination and polio. Now next is polio drops, which are given to children. It is also a vaccine, form of vaccine which is given as a drop to children. A worldwide campaign against smallpox has finally led to its eradication from most parts of the world, because all the people, they are much aware about that disease now, and this smallpox has been eradicated now. These days, vaccines are made on a large scale from microorganisms to protect humans and other animals from several diseases. So this was all about your microorganisms and how they help in your vaccination process. Next is how microorganisms increase soil fertility. So what do you mean by soil fertility? Soil fertility means when there is increase in the content of nutrients, useful nutrients and mineral ions in the soil. So how its fertility is increased? Some bacteria and blue-green algae, they are able to fix atmospheric nitrogen to enrich the soil. Enrich means to increase the fertility of soil with nitrogen and increase its fertility. These microbes, they are commonly known as biological nitrogen fixers, because they are fixing nitrogen from the atmosphere. You can see the different organisms such as bacteria, blue-green algae, which are the nitrogen fixers. These are the structures which appear under a microscope, because these you can't see with the naked eye. Next is your activity. Now you have to take 2 pots and fill each pot half with soil. Mark them as pot A and pot B. Put plant waste in pot A and things like polythene bags, empty glass bottles and broken plastic toys in pot B. Now put the pots aside. Observe them after 3-4 weeks. Did you find any difference in the content of the 2 pots? If you find the difference, then what is the difference? So this is an easy experiment you can perform at your home, and you have to do observation then and find the result. So what will you observe? You will find that the plant waste in pot A has been decomposed. Why it has been decomposed? Due to action of microorganisms and on the plant waste, on the organic waste, inorganic waste. So the plant waste has been converted into manure by the action of microbes. Now what is manure? You all might be aware of it. Manure are the substances which helps in the increase of fertility of soil. So these nutrients released in the process could be used by the plants again, and in this way the fertility of soil can be increased due to the presence of manures. Do you notice that in the pot B, the polythene bags, empty glasses and the bottle's broken toy parts did not undergo any such change. So why these substances do not undergo a change? Because on these substances microorganisms cannot act, and so that's why they will not convert into manure also. So if there will be no manure, then fertility of the soil will not be able to increase. So this is a easy observation which you can do and perform this activity at your home also. So this was all about your useful microorganisms. I hope you all have understood very clearly. So in our next video, we will study the rest part of this chapter. We will take into account the disease causing microorganisms, that is your harmful microorganisms, and how they cause diseases in plants and human beings, and we will discuss the various diseases with examples of microorganisms which are involved, and we will complete our chapter.